Good afternoon, classmates. I'm Gillen Joy Ortiz, and here with me is Nicole Cruz, and we are going to discuss family Sarcocystidae. So we have here its scientific classification. It is under phylum Amphicomplexa, class Sporozoa, subclass Coccidia, order Eucoccidida, suborder Emerina, family Sarcocystidae, and for its genus, we're going to focus on Sarcocystis and Toxoplasma, though it also have Besnotia and Neospora. So, family Sarcocystidae are heterocinus. Di ba yung life cycles of parasites can be further divided into two categories, which are the monocinus or direct life cycle or heterocinus whereas this parasite belong, meaning they are requiring or infesting more than one host to complete the life cycle. Parasites with indirect life cycle or itong ang heterocinus are characterized by two host stages which require a definitive host and an intermediate host. Also, please take note that both intermediate and definitive hosts are vertebrates. And with regards to the predatory prey life cycle, producing oocysts following syngamy, oocysts with two sporocysts, each with four sporozoites in the intestine of definitive host, are going to be more elaborated as we go with the life cycle of every genus. So let's proceed to our first genus under family Sarcocystidae, Sarcocystis. So, species that belong to this genus are causing disease called sarcocystosis, which is a zoonotic disease. Di ba kapag sinabing zoonotic, nakakahawa sa tao at animals. So, it can either be from animals to humans or from humans to animals. Genus Sarcocystis is the last generation merons found in the striated and the heart muscles, but rarely in the brain of the intermediate host. So, sarcocyst, ito yung large intramuscular cyst ng protozoan of the genus Sarcocystis. And also, sarcocyst is ordinarily divided into compartments by thin internal septa. So, by the word itself, septa, we already know that it is a dividing partition. Sarcocysts, depending on the species and stage of development, may either be microscopic or macroscopic. It contains initially the metrocytes, ito yung round-free merozoites, which mature as bradyzoites. And wall in some species has spine-like protrusion or radial spine seen in certain cysts of hosts called cytophaneres. So, Sarcocystis infects humans and animals worldwide. Human and carnivores act as the definitive host, while herbivores and omnivores act as an intermediate host. There are several species of Sarcocystis, but we will just be discussing about two species to which humans act as the definitive host, namely Sarcocystis hominis and Sarcocystis soi hominis. So, this is the life cycle of Sarcocystis species. Actually, yung pinapakita dito is yung Sarcocystis cruzi. So, yung final o yung definitive host nito ay dog, tapos yung intermediate host naman niya ay cattle. So, in the small intestine of the definitive host, namely the carnivores and humans, the microgamete fertilizes the macrogamete, forming unsporulated oocysts. These unsporulated oocysts undergo sporulation even inside the small intestine of the definitive host, forming mature or sporulated oocysts that describe as oocysts containing two sporocysts, each sporocyst containing four sporozoites, so a total of 8 sporozoites in one mature oocyst. And these oocysts may disintegrate in the intestine, liberating individual sporocysts. So what is passing the feces of the definitive host? Therefore, the diagnostic stages are both the sporulated oocyst and sporocyst. These sporulated oocysts and sporocysts are the stages that contaminate the environment like the grassland. So these are the stages that are infective too and being ingested by the intermediate host that as we can see in the picture it is the bovine. Once ingested, sporozoites within the sporocyst are liberated in the small intestine, penetrating the intestinal mucosa, then reaching the blood vessels. Now, in the blood vessels, sporozoites undergo three generations of synsogamy, forming three generations of merozoites respectively. And the third generation merozoite penetrate the muscle, transform to become non-infectious metrocyte. These metrocytes insist the muscle undergo asexual reproduction and further development to become mature, slowly dividing infectious bradyzoites contained within sarcocysts. The meat or muscle infected with sarcocysts containing bradyzoites is ingested raw or undercooked by the definitive host, which may be human or carnivores. Once ingested in the intestine of the definitive host, Sarcocyst releases active bradyzoites, infecting intestinal mucosa and become micro or macrogamete. The male macrogamete fertilizes the female macrogamete forming an unsporulated oocyst. And the cycle repeats and continues. This is another way of presenting the life cycle of the Sarcocystis species, showing the different diagnostic and infective stages, depending on the type of infection, which we'll be discussing later. So, sporocysts and oocysts are the diagnostic and infective stages, and bradyzoites within sarcocysts as diagnostic and infective stages as well. Now let's move on to pathogenesis. So Sarcocyst species are generally asymptomatic and is cleared spontaneously even without antiparasitic treatment. Nevertheless, if symptomatic, the infection is called sarcosporidiosis or sarcocystosis, which has two forms, the intestinal form and the muscular form. Intestinal form happens when human ingests sarcocysts containing bradyzoites, while muscular form happens when human ingests oocysts or sporocysts containing sporozoites. So this table shows the difference between muscular form and intestinal form of Sarcocystis infection. For muscular form, it happens when human ingests contaminated food or water with oocysts or sporocysts containing sporozoites, while intestinal form happens when human ingests raw or undercooked meat with sarcocysts containing bradyzoites, as I mentioned in the previous slide. And muscular form can be diagnosed by biopsy showing sarcocysts containing bradyzoites, while intestinal form can be diagnosed by demonstration of oocysts or sporocysts in the feces. And vascular form can be treated with cotrimoxazole, furazolidone, and albendazole and steroid as adjunctive therapy to decrease inflammatory response upon giving antiparasitic drug, while intestinal form is self-limiting and doesn't warrant any treatment at all. So, sarcocystosis is diagnosed in the intermediate host by histological examination, and infection in the definitive host can be demonstrated by fecal examination for oocysts. So, for diagnosis, Sarcocyst species can be diagnosed by stool examination, either by direct fecal smear or fecal flotation wet mount, which may show mature or sporulated oocysts described as containing two sporocysts. So, yung nasa taas na picture, as you can see, makikita natin dun yung dalawang sporocyst and kada sporocyst ay nagko-contain ng teardrop-shaped sporozoites which is apat na sporozoites. Or pwede din namang stool exam may show individual sporocysts o yung makikita natin dito sa picture below. So diagnosis can also be done by biopsy of infected muscles stained with hematoxylin and eosin. So one way to differentiate Sarcocystis soi hominis and Sarcocystis hominis is to check the cyst. If it can be seen by your naked eye, so macroscopic, it is Sarcocystis soi hominis. Otherwise, if it is microscopic, it is Sarcocystis hominis. So the photo above shows a muscle showing macroscopic sarcosis. So this is most likely Sarcocystis soi hominis. On the other hand, the other photo shows microscopic sarcosis. So this is most likely Sarcocystis hominis. So these are the three generations of merozoites of Sarcocystis species. And this picture shows you the sarcocyst containing several metrocytes and sarcocyst containing mature bradyzoites, which is the infective stage to human. For the prevention and control, since intermediate hosts acquire their infections by ingesting sporocysts, this could be prevented by proper disposal of definitive host feces and keeping the prey animal from predators. And definitive host becomes infected by eating prey animals on their meat. We can also do thorough cooking or freezing meat to kill bradyzoites or boiling water to kill oocysts or sporocysts containing sporozoites. And as of now, there is no approved vaccine yet.
So let's move on to the second genus of the family Sarcocystidae, which is the genus Toxoplasma. So here in the genus Toxoplasma, there is only one known species, which is the Toxoplasma gondii. So in the background, the Toxoplasma gondii has a worldwide distribution. It infects humans and many species of animals. This generally manifests as parasitic disease in immunocompromised individuals. So if we say immunocompromised individuals, it is the HIV patients and AIDS patients. So here, the cats act as definitive host, while the other animals like mouse, birds, pigs, cows, and sheep acts as an intermediate host, and humans act as an accidental host. So let's move on to the life cycle of the genus Toxoplasma. So the red one is the life cycle in the definitive host, which is the cat. And this one, the blue one, is the life cycle of the Toxoplasma gondii involving humans as their accidental host. So let's start with the definitive host. So in the intestinal cells of the definitive host, the male microgamete fertilizes the female macrogamete, producing an unsporulated oocyst that has been shed in the feces of cat. So this unsporulated oocyst undergoes sporulation in the external environment. And the sporulated oocyst is ingested by the intermediate host, which it can be the mouse or birds. And inside the body of the intermediate host, the sporocyte become the tachyzoites. So these tachyzoites localize in the neural and muscle tissue and insist as bradyzoites. So from sporozoites, it will become tachyzoites. And then it will insist as bradyzoite in the muscle tissues. If this insisted bradyzoite in the muscle is ingested by the cat, they become micro and macrogamete in the intestinal cells of the cat, and the cycles of the definitive host continues. But also, it is possible that the cat will directly ingest the sporulated oocyst. And now, let's move on to the life cycle in humans as an accidental host. So there are different ways on how humans can get the Toxoplasma gondii infection. The first one is the sporulated oocyst will contaminate the food we eat or the water we drink that will be consumed later on by the humans. So in this route, the infective stage is the sporulated oocyst. The second way is when the sporulated oocyst is eaten by the intermediate host like pigs or sheep. Then the sporulated oocyst will become encysted as bradyzooites in the muscle of the intermediate host. In that way, the humans can get this parasite by eating raw or undercooked meat of the intermediate host. So in this route, the infective stage is the encysted bradyzooite. So like for example, when we buy a barbecue and yung barbecue na yun is undercooked, then if yung pig na yun pala is infected with the Toxoplasma gondii, then the human that will eat the barbecue that is undercooked, then the human can get the Toxoplasma gondii. Then the third way is when the blood that will be transfused is infected with the Toxoplasma gondii, it can transfer in the other human. In this route naman, the infective stage is the motile tachyzoites. So when we need a blood transfusion and that blood is infected with the Toxoplasma gondii, it is possible that we can get the Toxoplasma gondii also. And then the fourth way is when the infected mother is transferred in the infection in the developing fetus. So in the transplacental transmission. So in this route, the infective stage is the motile tachyzoites. So once na yung mother is infected with Toxoplasma gondii, so pwede niyang matransfer yun sa kanyang anak. And then it is called as transplacental transmission. And the last route is when the infected solid organ or the bone marrow is transplanted to the recipient host. In this route naman, the infective stage is either encysted bradyzooites or motile tachyzoites. So like for example, if the patient has a leukemia and he or she needs a bone marrow transplant, then that bone marrow transplant is infected with the Toxoplasma gondii, then it is possible that the patient that will need that will need bone marrow can be infected with the Toxoplasma gondii also. So the infective stages of the Toxoplasma gondii to humans are the sporulated oocyst, encysted bradyzooites, and motile tachyzoites. So the tachyzoites enter in the blood circulation. Once they recognized by the immune system of the human host, they will become bradyzooites and encysted themselves in various organs to protect them from the immune system of the human host. So when we said encysted, the bradyzooite will have a cyst or a wall para mas maprotektahan niya yung sarili niya sa immune system ng isang tao. So once the immune system of the host goes down, it will detect by the parasites. And they will exist, releasing the motile tachyzoites, which will go into the different organs of the body, causing disseminated toxoplasmosis that will later on will be discussed. And then, the Toxoplasma gondii infection can be diagnosed by tissue biopsy showing encysted bradyzooites, blood smear showing tachyzoites. So like in the picture, the one with have arrow is the tachyzoites. It is crescent in shape. And then third one is the serum or cerebrospinal fluid for serologic testing. So the three forms of the Toxoplasma gondii are the oocyst, tachyzoites, and bradyzooites. So let's start with the oocyst. Oocysts, these are the oocysts found in the stool of the definitive host, which are the cats. So it is similar in appearance to those of the Cytoisospora but relatively smaller. It contains 2 sporocysts. Each sporocyst contains 4 sporozoites, making it a total of 8 sporozoites all in all. So, kaparas lang siya ng report ni Gillen kanina. The second form is the tachyzoites. So, these tachyzoites has a single central nucleus, crescent in shape, and highly motile and rapidly dividing. So as you can see in the picture, it is crescent and the nucleus of the tachyzoite is in the center. And lastly is the bradyzoites. So the bradyzoite, which is similar to the tachyzoite but the single nucleus is located posteriorly, since the tachyzoite which nucleus is located in the center. And also, there are hundreds or thousands of the bradyzoites enclosed in a cyst that is slowly dividing. So the difference between the tachyzoites and bradyzoites is, in the tachyzoites, the nucleus found in the center, while in the bradyzoites, their nucleus is found in the posterior part. And also, in the tachyzoites, it is highly dividing and motile, while in the bradyzoite it is slowly dividing. So let's move on to the pathogenesis. So in the pathogenesis, the Toxoplasma gondii is generally asymptomatic to the immunocompetent individuals. When we say immunocompetent individuals, malakas yung immune system nila. And then the second one is it causes toxoplasmosis in the immunocompromised patients. When we say immunocompromised patients naman, it is the HIV patients and AIDS patients. So we can find in the encysted bradyzoites in different parts of the body like the skeletal muscle, myocardium which is the heart, brain, and eyes. In the acute infection, the patient may experience fatigue, lymphadenitis, fever, chills, headache, myalgia. On the other hand, the chronic infection, the patient may experience encephalomyelitis, myocarditis, hepatitis, retinochorditis with subsequent blindness. So in the disease, first one is the congenital toxoplasmosis. So the congenital toxoplasmosis may happen to mother-to-child transmission that is severe and fatal, with the classic triad of hydrocephalus, intracranial calcification, and chorioretinitis. So hydrocephalus means there is a water in the brain. Intracranial calcification means there is a calcification within brain parenchyma or the vasculature. And chorioretinitis, it may happen in the retina of the eyes. Then the other manifestations of the congenital toxoplasmosis are the microcephaly, mental retardation, convulsions, psychomotor abnormalities, severe visual impairment, or blindness. The second disease is the cerebral toxoplasmosis. It happens in the immunocompromised patients like HIV and AIDS, manifesting as chronic headache, focal neurologic deficits, seizures, fevers, altered mental status, and lethargy. So in this imaging picture, we can see multiple ring-enhancing lesions which is pointed by the red arrows. So let's move on to the diagnosis of the Toxoplasma gondii infection. The laboratory diagnoses are done by, first one is the demonstration of the tachyzoites in the blood or encysted bradyzoites in tissues. Or we can also do a polymerase chain reaction, or serologic testing that employs antibody detection, which is the immunoglobulin M and the immunoglobulin G, or the Sabin-Feldman dye test. So what is Sabin-Feldman dye test? Sabin-Feldman dye test, it uses methylene blue as a stain of the cytoplasm of the Toxoplasma gondii. The laboratory-grown Toxoplasma gondii is incubated with the patient's serum at 37 degrees Celsius for 1 hour. So in here, the interpretation of result is if the patient has antibodies against Toxoplasma gondii, the antibodies and complement lies the plasma membrane of the Toxoplasma gondii. Hence, positive result is no Toxoplasma gondii being stained with methylene blue. But if the patient has no antibodies against Toxoplasma gondii, the laboratory-grown Toxoplasma gondii are not being destroyed after addition of the patient's serum. Hence, negative result is presence of intact Toxoplasma gondii being stained with methylene blue. Meaning to say, when the patient have antibodies against Toxoplasma gondii, the cytoplasm or plasma membrane of the Toxoplasma gondii doesn't have stain. But if the patient doesn't have antibodies against the Toxoplasma gondii, the Toxoplasma gondii cytoplasm has stained. So like here in the picture. Kapag sinabi natin, kapag yung patient is meron siyang antibodies na nabuo, yung cytoplasm ng Toxoplasma gondii, wala siyang stained. While kapag yung patient naman is walang antibodies na nabuo, yung Toxoplasma gondii is merong stained yung cytoplasm nila. So now, let's move on to the treatment. So pyrimethamine plus sulfadiazine with folinic acid. If the patient is pregnant, in the first 4 months, we can give pyrimycin, and in the succeeding months, pyrimethamine plus sulfadiazine with folinic acid. So let's move on to the prevention and control. First one is the avoidance of contact with cat feces. Pregnant women should avoid contact with cats as well as the litter box of the cat. And then, meat and eggs should be well cooked. Unpasteurized milk should be avoided. Laboratory workers should be careful in handling parasites. Then, veterinarians may administer Toxoplasma vaccines in the cat. This vaccine is originally made for sheep to reduce effects of the Toxoplasma gondii infection like the embryonic death. In cats, it is given to inhibit sexual development of Toxoplasma gondii.
Let's now proceed to our quiz. For question number 1, what is the last generation neurons found in the striated and heart muscles of the intermediate host? Again, what is the last generation neurons found in the striated and the heart muscles of the intermediate host?
Number 2, what are the classic triad? Again, what are the classic triad?
For question number 3, true or false? Intestinal form happens when human ingests sarcocysts containing bradyzoites. For number 3, true or false? Intestinal form happens when human ingests sarcocysts containing bradyzoites.
For number 4, true or false? Positive in antibodies, no stain. Negative in antibodies, have stain. Number 4, true or false? Positive in antibodies, no stain. Negative in antibodies, have stain.
For question number 5, true or false? Muscular form is self-limiting and doesn't warrant any treatment at all. For question number 5, true or false? Muscular form is self-limiting and doesn't warrant any treatment at all.
Number 6, true or false? The Toxoplasma gondii vaccine is originally made for cat to reduce effects of Toxoplasma gondii infection like embryonic death. Number 6, true or false? The Toxoplasma gondii vaccine is originally made for cat to reduce effects of Toxoplasma gondii infection like embryonic death.
For question number 7, what do you call the radial spine seen in certain cysts of hosts? For question number 7, what do you call the radial spine seen in certain cysts of hosts?
For question number 8, the third way of the life cycle of Toxoplasma gondii in accidental host is when the blood that will be transfused is infected with the Toxoplasma gondii, it can be transferred in the other human. In this route, what is the infective stage that has been mentioned? The third way of life cycle of Toxoplasma gondii in accidental host is when the blood that will be transfused is infected with the Toxoplasma gondii. It can be transferred in other human. In this route, what is the infective stage that has been mentioned?
For question number 9, how many sporozoites each sporocyst have? For question number 9, how many sporozoites each sporocyst have?
Last question, number 10. Give only one diagnostic specimens and stages of the Toxoplasma gondii. Number 10. Give only one diagnostic specimens and stages of the Toxoplasma gondii.